So I found this article very interesting because I had just recently given a presentation over research about cannabinoids and their utilization on cancer cells. So this article was really interesting because it kind of piggybacked off of that. But to get started, it said a main pillar of innovation in cancer therapy has been an improved understanding of the underlying tumor biology. The knowledge has led to the development of small molecules and antibodies targeting key proteins of oncogenic signaling pathways. While these agents have improved survival for selected cancer entities, for many others, treatment options are still limited and resistance mechanisms are poorly understood. However, while most genetic variations of cancer genomes have been structurally characterized, much less is known about the function of many mutated genes. This particularly applies to the long tail of molecular alterations, which are currently found only at low frequency. Recently, genome engineering was greatly accelerated by the development of the CRISPR Cas9 technologies. Since its first use as a genome editing tool in 2013 in mammalian cells, the toolbox of CRISPR-Cas9 has been continuously expanded, enabling not only the modification of the genomic sequence of cells and organisms, but also the introduction of epigenetic and transcriptional modifications. CRISPR-Cas9 can be used to explore the non-coding genome of cancer. Furthermore, we can describe novel in vitro and in vivo cancer models that can be engineered by the CRISPR-Cas9 as well. CRISPR stands for the clustered, regularly interspaced short palindromic repeats, which are short repeated sequences of DNA nucleotides found within the genome of prokaryotes. Palindromic repeats are the same sequencing from 5' to 3' going both ways. So 5', 3', 5' to 3', it's the same back and forth. The example used in the video was race car. Race car forward and backwards is still race car. Anyway. CRISPR and its CRISPR-associated proteins, Cas, are linked to the adaptive immunity targeting foreign viral DNA. Mechanistically, two distinct RNAs, the CRISPR targeting cRNA and the transactivating RNA, the TRACR RNA, activate and guide Cas proteins to bind viral DNA sequences, which are subsequently cleaved. A subgroup of the CRISPR systems, the type 2 system, relies on a single Cas protein to target a defined DNA sequence and is therefore particularly attractive to be used as a tool for genome editing. In most cases, CRISPR interference systems rely on dCas9 fusions with Crab repressor domain to downregulate gene transcription. Interestingly, dCas9 Crab can also modify enhancer regions by changing the methylation status at the target site, thereby repressing gene transcription. In cancer cells, transcription is deadly because it keeps copying and forming new cancer cells, and they never go into apoptosis or they never die. Recently, Cas9 variants have been engineered that induce specific signal nucleotide base changes. These base editors do not introduce DSBs, but rely on Cas9 nicases coupled to cytidine deaminase domains to introduce targeted transitions from C to T or G to A. The lesson learned from these experiments is that off-targets share sequence similarities with the target sites, but the precise bioinformatic prediction of all off-targets is not possible based on the currently available data. The concept of gene essentiality can be narrowed down to gene regions encoding essential protein domains, as opposed to RNAi that depletes transcripts of target genes. It is possible to disrupt specific protein domains using the CRISPR-Cas9 by targeting the corresponding genomic sequence. This screening approach, termed domain screen, was used to pinpoint protein domains that determine the essentiality of a specific gene, which can further facilitate drug development. A similar approach identified that AML cells lines harboring a mutation in the NPM1 gene, specifically depend on benign binding site of the MLL1 protein. Supporting this observation, pharmacological inhibition of the binding site has been pronounced anti-leukemic effect. Synthetic lethality occurs if a cell can compensate the functional alteration of gene A or B, but is unable to survive if gene A and B are simultaneously altered. Translated into cancer biology, this means that previously non-essential genes can become essential due to the loss of a specific tumor suppressor or the activation of an oncogene. The translation relevance of this concept was recently demonstrated by a clinical trial that showed higher sensitivity of breast cancer bearing a germline mutation of BRCA to PARP inhibition. Another major application of the CRISPR-Cas9 screening is the dissection of chemicogenetic interactions, which enables insights into how cancer responds to drug treatment. Combining pooled CRISPR screening with a drug as a perturbation can identify gene knockouts that act synergistically with or confer resistance to the agent. Disruption of tumor suppressors such as APC, TP53, and SM84, as well as genetic editing of oncogenes such as CRAS and PI3K, lead to organoid models of colon cancer in vitro. Of note, these cells were growing independently of any growth factors normally needed for the culture of untransformed organoids, therefore enabling phenotype-dependent selection of genetically modified organoids. Adeno-associated virus, AAV, are regarded as a potent tool as they are non-integrating, possess a high transduction efficiency, and are serologically compatible with a large fraction of the human population. Furthermore, the families of AAVs is characterized by a rich diversity of serotypes, which distinct tissue tropisms, enabling the selective targeting of different organs. And although both Cas9 expression and AAVs caused a humoral immune response, no extensive cellular damage was observed. The CRISPR-Cas9 provides a tool to manipulate non-coding regions of the genome and will accelerate the function exploration of a so far poorly characterized aspect of the cancer genome. The precise engineering of common and rare mutations of the CRISPR-Cas9 combined with advanced organoid culture will enable cancer researchers to copy the genetic sequence of carcinogenesis of many cancer entities. So, in short, the CRISPR-Cas9 has opened up the research and the ability to be able to attack the cancer cells without them being able to mutate to form a resistance against the drugs or the treatment or whatever it is. A lot of times in cancer patients, that's one of the main issues, is that their body cannot handle the treatment that they're getting, or that their body denies the treatment, or like in this case, the tumor cells of the cancer have mutated to be just kind of like have that drug put into them and then kick it out and keep going. So with this, I do believe that there will be positive outcome of this in the future as more studies and trials keep going on for cancer patients. But yeah, overall, I really like this one, and I'm excited to look back in a couple years or even in a couple months and see if this has accelerated any.